Hello everybody, welcome back to Darwin Academy. This is Varsha Kulkarni. Today let's learn something more about Golgi apparatus. Unlike every other time, I have only drawn the cell organelle here. Every other time you would have seen me drawing a cell with all the cell organelles, but today I decided to draw only the Golgi apparatus along with a little bit of information here. I hope this would be useful. And also, I would want to keep today's topic short because not much is discussed in your grade 9 NCERT curriculum. We'll be looking at brief history of Golgi apparatus. Let's talk a little bit more about its discovery and all those things under that subheading of brief history. Followed by let's study the structure of Golgi apparatus and let's move to its functions, and before that we will see where is it located. Alright, this will be the flow of today's topic. With that being said, let's get started. I hope all of you are ready with your sheet of paper and pencil or pen to scribble down the points. Today's cell organelle that we are going to discuss is called as Golgi apparatus. It was known by some other name before 1910. You need not know what name it was called by before 1910, but from 1910 onwards, the term Golgi apparatus has been used. So, a scientist by name Camillo Golgi, who was from Italy, studied the nerve cells extensively, and within these nerve cells, he was able to spot layers or stacks of these vesicles that he called them Golgi apparatus. So it was in 1898 that Camillo Golgi first discovered the Golgi apparatus. The name Golgi apparatus that we use of today was first used in 1910. Camillo Golgi won a Nobel Prize along with another scientist for developing a staining technique to stain the nerve cells. The understanding that we have of the nerve cells is because Camillo Golgi was able to stain these nerve cells, and as a result we can study them at depth today, right? So for that he was awarded the Nobel Prize, but not for the discovery of Golgi apparatus. Let me quickly repeat a few important points about the brief history and who discovered it and all those things. So, Golgi apparatus is the cell organelle that we are going to discuss today. It was discovered in 1898 by Camillo Golgi. The term Golgi apparatus was being used from 1910 onwards. Camillo Golgi won the Nobel Prize along with another scientist for his method of staining the nerve cells. Now, where are these Golgi apparatus, which is also known as Golgi bodies or Golgi complex, is found? Location. So, Golgi apparatus is found within all eukaryotic cells, meaning those cells that are well developed, which have a clear, well-defined nucleus, nuclear membrane, and membrane-bound organelles, right? So, we find Golgi apparatus in all eukaryotic cells, and they are within the cytoplasm and very close to the endoplasmic reticulum. By the way, if you have not watched the endoplasmic reticulum video that I have put out very recently, please go back and check that out. I have discussed about endoplasmic reticulum in that video in detail. So Golgi apparatus is found very close to endoplasmic reticulum within the cytoplasm, and they are present in all eukaryotic cells. Now, how is the structure? Before going on to the structure, let me ask you a quick question. What do you know or what is meant by a vesicle? V-E-S-I-C-L-E. Vesicle is, I would like you to take you a guess or two. If you think you know, try and answer it. Write it down in the piece of paper that you already have. Okay. Vesicle is a small oval or roundish kind of a structure within which there is some fluid stored. right? The fluid is stored within these oval structures which are covered by membrane, and such structures which have fluid within them and covered by membrane are called as vesicles. The Golgi apparatus has flat vesicles, or compare the vesicles to a sac, a round sac. Imagine if this round sac is flat in nature. That is how the Golgi apparatus will be. So, these flat sacs are called as cisterns, singular, right? If there are many cisterns placed one above the other, then it is called as cisternae. So Golgi apparatus has many such cisternae stacked one above the other. Got it? You should have understood what vesicle is, what is a cistern, and what is a cisternae. Let me repeat that quickly for you. Any round oval structure which is covered by a membrane and has fluid within itself is called a vesicle, right? I will consider, or I'll always remember the vesicle like a round sac. Now, imagine if the sac is flat in nature, right? I hope you are able to visualize the flat sacs, right? That is how the flat sac cistern is present, right? These cisterns are laid one on top of the other, just like this, to form the Golgi apparatus, right? Many cisterns are called as cisternae. I hope you have understood the point. With that being said, you should also know that the Golgi apparatus is connected with the membranes of endoplasmic reticulum. I hope this much is clear. You have understood the brief history of endoplasmic reticulum, the location and its structure. Moving on to the last bit of today's topic, that is the functions of a Golgi apparatus. Since these are sac-like structures, they are involved in storage, they are involved in modification, packaging, and also distribution and dispatch of materials that are synthesized by the endoplasmic reticulum. That is the first and important job that the Golgi apparatus is assigned with. Remember that endoplasmic reticulum is involved in the synthesis of proteins and lipids. So once they are produced from the endoplasmic reticulum, since the Golgi is very much closely connected with the endoplasmic reticular membrane, right, it is involved in storage, manufacture, packaging and distribution of these materials into the cell or within the cell and also outside of the cell, right? And also the Golgi apparatus is entrusted with another job, that is to manufacture the lysosomes. We'll be talking about lysosomes in the upcoming videos. In the previous video of endoplasmic reticulum, I have told you that the lysosomes have digestive enzymes which are being synthesized by endoplasmic reticulum. Since the digestive enzymes, the component of the lysosome, is manufactured by the endoplasmic reticulum, the actual structure of the lysosome is made by Golgi apparatus. I think with this, I'll be able to conclude today's topic. But before winding up, let me quickly whiz through the topic. We have discussed about the cell organelle called Golgi apparatus. We know that Camillo Golgi discovered them in 1898 when he was studying about, or when he was studying extensively the nerve cells. We also know that the Golgi apparatus or the Golgi body or the Golgi complex is made up of flat sac-like structures which are called as cisternae, and these cisternae are stacked one above the other. They are also connected with the membranes of the endoplasmic reticulum. The Golgi apparatus is present within all eukaryotic cells, right, within the cytoplasm, and they are attached very close to the endoplasmic reticulum because they are involved in storage, modification, then packaging and distribution of the materials that the endoplasmic reticulum synthesizes within the cells, and they are also involved in transport or distribution of these materials outside of the cell as well. Finally, to conclude, Golgi apparatus is also involved in the production or manufacture of lysosomes. I think I'll be concluding today's topic. I hope you have all understood. Please do like, share, and subscribe. Thank you. See you all in the next one.